Hello my dear students, today we'll start transport of nutrients in plants. This process occurs gradually. This is a physiological process in plants and it occurs in three successive steps. The first one is the roots take up water from the soil, then water is drawn up the stem to the leaves, and then thirdly water evaporated from the leaves. Here the two types of plant tissue, xylems and phloems, are involved to conduct the process. Transport in plants, basically all living organisms requires the movement of essential substances such as water, minerals, respiratory gases and nutrients. In case of unicellular organisms like amoeba, paramecium or any other bacteria, the transport occurs through diffusion and osmosis. The single cell is in direct contact with the surrounding environment or ambience. But in case of multicellular organisms, particularly in plants, a complex transport tissue system is needed due to the large body size and tissue differentiation. Plants use specialized vascular tissue for transport, like xylem transport water and mineral salts. They transport unidirectional, that is from roots to stem and leaves. Phloem, it transport organic food materials, mainly sucrose. Is here movement is a bidirectional process from leaves to other parts and vice versa as needed. Both xylem and phloem together form a continuous vascular system. It ensures smooth distribution of substances throughout the plant. It supports growth, development and survival of the plant. Here the diagram shows the xylem has water and minerals and transport. It conduct water and mineral transport, and one-way flow of sap occurs here. Thick cell wall made up of lining is present in xylem, and cells having no end walls between them. But the phloem cells have end walls and perforations between them. Cell wall is made up of cellulose. There is a two-way flow of sap, and water and food are transported through the phloem. Here the diagram show the role of xylem and phloem. Xylem conducts water and mineral transport from roots to the leaves, and then phloem conducts both food prepared by leaves to the storage organs. Water and minerals are transported, and food prepared by leaves are transported throughout the plants. And here the tissues of the leaves are adapted for gaseous exchange. Water reaches leaves for photosynthesis. Tissues are adapted to conduct water and minerals. Stem is here, the water in soil, and root here. Cells of roots are adapted to absorb water and minerals from the soil. Xylem and phloem, there is a comparative discussion. Xylem transport water and minerals. Phloem transport food in the form of sugar, and it store food as storage sucrose. Xylem is responsible for one way or upward flow, and phloem is responsible for two-way flow. And in case of xylem, transport is the bulk flow due to negative pressure, but in phloem transport is by osmosis or turgor pressure. And xylem consists of hollow dead cells and there are no end walls are there. Phloem consists of live cells with no nucleus and perforated end walls are present there. In xylem, they form center of the vascular bundle, but outside the edge of the vascular bundle phloem is present. Xylem is rich in lignin, which supports the plant. Phloem also transports amino acid, mRNA and hormones. There is a diagram of both plant cell, label diagram of both plant cell and animal cell. Plant cells have ribosome, Golgi vesicle, smooth endoplasmic reticulum where no ribosome, nucleus, rough endoplasmic reticulum where ribosomes are present on the surface, large central vacuole, amyloplast. There is cell wall is an essential feature of plant cell which is absent in animal cell. Then cell membrane, Golgi apparatus, vacuole transport, chloroplast, graphite crystal, Druss crystal, mitochondria, cytoplasm. But in case of animal cell, there is pinocytic vesicle, lysosome. Lysosome is absent in plant cell. Golgi vacuoles, rough and smooth endoplasmic reticulum, plasma membrane, which is the most outer layer of the animal cell, where plant cell where cell wall is absent. Mitochondria, Golgi apparatus, nucleus, nucleolus, centrioles, microtubules, ribosomes are present on the cytoplasm of the animal cell. So this is the difference. Plant cell have cell wall, but animal cell have no. Our cell membrane is the outermost layer. Cell membrane is present both in plant cell and animal cell, but in plant cell it is located just beneath the cell wall. Chloroplast, plant cell has chloroplast, but animal cell has no chloroplast. Plant cell has one large central vacuole, animal cell has small or absent vacuoles. Both plant and animal cells have nucleus and nucleolus. Both of them have endoplasmic reticulum, Golgi apparatus, Golgi vesicles, and ribosomes, mitochondria, cytoplasm. protoplasms, but plants have only plastid for the sake of photosynthesis and food storage and formation of colors due to which help in pollination. Animal cell has no plastid. Plant cells has dross and graphite crystals, which is specific to plant cells for storage and defense, but animal cells have not. Plant cells have no pinocytic vesicle, but animal cells have pinocytic vesicle, which is involved in endocytosis. Plant lysosome is rare or absent in plant cell, but it is present in animal cell. Centrioles are absent in plant cell, but centrioles are present in cell division. Involved in cell division, microtubules are less prominent and not shown, but animal cell ক্ষেত্রে microtubule is present there. But in case of plant cell division, there is a MTOC, microtubule organizing center, plays an important role for the spindle formation. Here the types of plant tissue. Plant tissue is basically two types: permanent tissue and meristematic tissue. Permanent tissue where cell division has been stopped. It is of two types: simple permanent tissue, which are made up of single type of cells like parenchyma, collenchyma and sclerenchyma, and which at permanent tissue are made up of two or more than two types of cells, are called as complex permanent tissue. There are two types, xylem and phloem, and xylem includes tracheic vessels, xylem parenchyma and xylem fiber, but the phloem consists of sieve tube elements, companion cells, phloem parenchyma and phloem fiber. Meristematic tissue always undergoing cell division. They are present in the apical meristem. They are present in the apical portion, which is called as apical meristem. Some of them are present in the intermediary region, they are called intercalary meristem, and some are located in the lateral portion, they are called as lateral meristem. So xylems, now we'll discuss the structure and characteristics of xylem. And xylem is a complex permanent tissue in vascular plants, responsible primarily for the transport of water and mineral salts from roots to other parts of the plant. It also provides mechanical support. Main functions of xylem: it conduction of water and minerals from the roots to aerial parts, the mechanical support due to the presence of lignified elements. Procambium is a type of primary meristem, meaning it appears early in the plant's life and especially in growing tips of roots and shoots. It is responsible for forming the initial vascular tissues, primary xylem and primary phloem, that help young plants conduct water and fluid. Think of it as the starter system for plants' plumbing network. As the plant grows older and thicker, especially in woody plants, it needs more support and more pathways to transport nutrients and water. Here the vascular cambium comes into action. It is a secondary meristem that appears later and helps in the plant's thickening or gut. It produces secondary xylem or wood on the inner side of the secondary phloem on the outer side. The vascular cambium often originates from the procambium in the vascular bundle and also from the parenchyma cells between bundles or interfascicular region. So you can say procambium gives rise to the vascular cambium, and the vascular cambium continues the job but on a larger scale as on the plant matures. Xylem components. Xylem basically consists of four main types of cells. Tracheids. The structure of tracheids, they are elongated tapering cells have with thick and lignified walls. They are dead at maturity and they lack protoplasm. They have broader pits present for lateral conduction. They are predominant in gymnosperms, but they are also present in angiosperms. Their function is conduct water and minerals vertically and laterally. They provide mechanical strength. Vessel structure. Long tubular structures formed by end-to-end fusion of vessel elements. The end walls are perforated or completely dissolved, which has perforation plates. The dead cells with lignified secondary walls. They predominantly found in angiosperms. They are absent or rare in gymnosperms. Efficient water conduction due to continuous tube-like structure. Xylem parenchyma, here the special feature, the only living component of the xylem tissue. Living cells with thin walls. They may be elongated or polygonal. Their function is storage of starch, fats, tannins and other substances. They assist in lateral transport of water and minerals. Xylem fibers, which are the fibrous element, they are dead, thick walled, elongated and lignified cells. They are called as libriform fibers. Their function is to provide mechanical support. They do not participate in conduction process. Here the elements of xylem. Special features of xylems: all components except xylem parenchyma are dead at maturity. Lignin deposition provide rigidity and prevents the collapse under water tension. Unidirectional flow of water from root to shoot. It forms annual ring in secondary growth, which is useful in dendrochronology. Phloem. Structure and characteristics of phloem. Phloem is basically a complex permanent tissue in vascular plants, responsible for the translocation of organic nutrients, mainly sugars like sucrose, from the leaves to all parts of the plant, that is zinc. source and sink mechanism occurs here. Main function of phloem is transport organic solids, especially products of photosynthesis, the bidirectional movement of food, unlike xylem, which is unidirectional and also involved in signaling molecules and certain hormones transport. The origin primary phloem arises from the procambium, and secondary phloem is produced by the vascular cambium during secondary growth. Phloem components. Phloem is made up of living and non-living elements. Sieve tube elements or sieve tubes are elongated living cells arranged end to end, and the end walls are perforated, forming sieve plates. They lack nucleus at maturity but contain cytoplasm. Their function is main conducting elements for translocation of sugars. The transport occurs via mass flow driven by osmotic pressure. Companion cells, they are living and nucleated cells. They are connected to sieve tube elements via plasmodesmata. They are smaller than sieve tubes, lie adjacent to them. Their function assist in loading and unloading of sugars, help maintain the metabolic function of sieve tubes, involved in energy intensive transport, that is active transport. The phloem parenchyma is the living cells with a nucleus and cytoplasm. They are elongated with tapering ends. Their function is to storage of starch, resin, latex and other substances. They may also aid in or help in lateral transport of food. Phloem fibers or bast fibers are the only non-living component of phloem. Structure dates sclerenchyma cells with thick lignified walls. They are long, narrow and tapering at ends. They provide mechanical support to the plant. Special features of phloem: they contain both living and dead element, bidirectional transport, source or sink and vice versa based on needs. Sieve tubes and companion cells are functionally and structurally interdependent to each other. Phloem is very vital for growth, repair and storage in plants. There is a difference between xylem and phloem. Water and minerals are only transported in xylem, but phloem transport only water and food. The xylem conduct one-way movement, but phloem cells are end walls with perforation, so it involved in two-way movement. So in xylem there is no end walls with between cells and thick walls with steeped lignin. Xylem transport water and minerals. Phloem transport only organic food and water. Direction: unidirectional. Xylem transport. Phloem transport is bidirectional from source to sink and vice versa. Cell type: they are mostly dead cells at maturity and mostly living cells at maturity. Main elements: tracheids and vessels, sieve tubes and companion cells. Modes of absorption of nutrients in plants. Plants absorb nutrients mainly through their roots from the soil. This process occurs via different physical and physiological mechanisms depending on the nature of the substance and concentration gradients. Movement of molecules from a region of higher concentration to lower concentration. Characteristics are occurs along the concentration gradient. It is slow and non-selective process. It does not require energy. Example: it entry of gases like carbon dioxide into leaves, movement of mineral ions like chloride in the soil solution into the root hairs. Osmosis: special type of diffusion involving water molecules through a semi-permeable membrane form a region of higher water potential to the lower water potential. They have characteristics: it involves only water. Affected by solute concentration, it does not require energy. Example: absorption of water from the soil into the root hair cells. Passive transport: transport of substances across a membrane without the use of cellular energy is called as passive transport. It occurs along concentration gradient and can occur via simple diffusion or facilitated diffusion through transporter proteins. There no energy is required in this process. And movement of potassium nitrate through ion channels or carrier protein when soil concentration is higher. Active transport: movement of substances against the concentration gradient from low to high concentration using ATP or energy is called active transport. Its characteristics: highly selective and requires transporter protein or pump. The energy dependent process: uptake of mineral ions like calcium, potassium, in sodium, phosphate when their concentration is lower in the cell than inside the root cells. So diffusion requires no energy. Its gradient direction is high to low. It is no selectivity. Example: carbon dioxide comes into the leaves, and chloride uptake. It is an example of diffusion. In case of osmosis, no energy is needed. Gradient transport: high water potential to low water potential. There is no selectivity, and water into root hair cells. This is an example. But passive transport, it requires no energy. Gradient is high to low, selectivity sometimes, and the example of passive transport is the potassium uptake via ion channels. Active transport, it requires ATP or energy. Here the gradient direction is low to high and its selectivity is yes. So it is phosphate and nitrate uptake by root cells. So absorption of roots. Absorption by roots refer to the physiological process by which water and mineral nutrients are taken up from the soil and transported to other parts of the plant. Types of absorption হচ্ছে passive absorption driven by transpiration pull from leaves. It occurs due to osmosis, diffusion and capillary action. No metabolic energy or ATP is required. Main process in tall trees and during high transpiration. Active transport, energy dependent uptake of minerals and sometimes water. It involves ATP and carrier protein in plasma membrane, dominant when transpiration is low. Example at night in high humidity. Here the key features are the root hairs increases the surface area, thin walls are responsible for easy diffusion or osmosis, high cell sap concentration maintain osmotic gradient. The mycorrhiza enhances the mineral uptake by the root cells, and carrier protein enables selective absorption. So root hair zone is the primary site for water and mineral absorption. Meristematic zone actively divides but absorbs minimally water and salt. And the third one is the maturation zone differentiated to specialized cells including root hairs. Transpiration. Transpiration is basically a physiological process by which water in the form of vapor is lost from the aerial parts of a plant, mainly through the stomata of leaves but also through the cuticle and lenticel of stems. Types of transpiration, basically three types of transpiration are found there. Stomatal transpiration, here loss of water vapor through the stomata, which are the tiny pores found on the leaf surface, mostly on the underside of the leaves, accounts for more or less 80 to 90% of total transpiration. Cuticular transpiration, here water loss directly through the cuticle, the waxy non-cellular layer and covering leaf surface, around 5 to 10% of the total transpiration process. Lenticular transpiration, evaporation of water vapor through lenticels, which are the small pores present on the bark or woody stem on the plants. Here the contribution is very minor, that is less than 1% of total transpiration. Now we'll discuss about the absorption and transpiration. Absorption and transpiration in plants are closely interconnected processes that work together to maintain water and nutrient balance. As water evaporates from the aerial parts of the plant, mainly through stomata, it creates negative pressure called transpiration pull. This pull helps in drawing water upward from the roots through the xylem. To compensate for the water lost during transpiration, roots absorb more water from the soil. This not only ensures a continuous supply of water but also aids or help in the uptake of essential mineral nutrients. Thus, transpiration indirectly regulates and enhances water and mineral absorption in plants, supporting their physiological functions and growth. Ascent of sap is the basically the water tank is water that is absorbed from the soil by the root hairs gradually accumulate in the tissue of the cortex. Sometimes some part of this water is transferred by the aerial surface of the leaf. However, before the water can be transferred, it must reach the aerial surface against the downward pull of gravity. This upward movement of water and dissolved minerals is called as ascent of sap. Factors responsible for ascent of sap: root pressure, which is generated due to active absorption of mineral salts by the root cells. It creates a positive pressure that pushes water upward from the root xylem. It is most effective in small plants or during low transpiration periods, that is occurs at night. It is evident through guttation and exudation. Transpiration pull, it is caused by the evaporation of water from leaf surface, mainly through stomata. It creates negative pressure or suction force that pulls water upward from roots through the xylem. This is the main driving force for the ascent of sap is found in tall trees. Force of cohesion and adhesion. Cohesion, where water molecules stick together due to hydrogen bonding forming continuous water column, that is called as cohesion. But in case of adhesion, water molecules stick to the xylem vessel walls, preventing the column from breaking. Together these forces maintain the integrity of the water column as it pulled upward. Factors regulating transpiration: external or environmental factors. Temperature: higher temperature increases the rate of evaporation and hence transpiration. Too high temperature may lead to stomatal closure to prevent excessive water loss. Light, that is intensity and duration of light, is the important factor. Light stimulates stomatal opening and increasing transpiration during the day. More light causes more photosynthesis and more water demand, more transpiration. Humidity: high humidity reduces transpiration, that is less vapor gradient. Low humidity increases transpiration due to steep water vapor gradient. Wind air movement: wind removes the humid air around the leaf surface, increasing transpiration. Still air slows down the rate. Atmospheric pressure: lower pressure, that is example at high altitudes, enhances evaporation and transpiration. Internal or plant related factors. Stomatal regulation, opening and closing of stomata is the primary control point of transpiration, regulated by guard cells in response to light, carbon dioxide concentration and water status. Lip surface area and structure. Larger surface area indicates higher rate of transpiration. Thick cuticle and fewer stomata reduce the water loss. Cuticle thickness. A thick waxy cuticle reduces cuticle transpiration. Root hair uptake, adequate water absorption by roots support sustained transpiration. Here water stress leads to reduced stomatal opening. Types of plant. Xerophytes, desert plant have adaptation to minimize transpiration. Hydrophytes and mesophytes transpire more, so open stomata and closed stomata are there. So these are the internal or plant related factors responsible for transpiration. Advantages of transpiration: creates transpiration pool, generates a suction force that helps in the upward movement of water and minerals from the roots to leaves via xylem, facilitates cooling, evaporation of water from leaf surface cools the plant, protecting it from heat stress similar to sweating in human. The aids in mineral transport, helps in the movement of dissolved minerals from the soil to various parts of the plant. It maintains turgidity, it continuous water flow, maintains cell turgor, supporting structure and physiological processes like growth and stomatal function. They remove excess water, helps in disposing of surplus water absorbed by roots, especially when water intake exceeds metabolic need. Disadvantages of transpiration: excessive water loss can lead to wilting, reduced growth, or even plant death under drought conditions if water loss exceeds uptake. Reduced photosynthesis during water stress: stomata may close to prevent transpiration, which also limits carbon dioxide intake and reduces photosynthesis. Energy waste in dry condition: in arid region, water loss through transpiration can be a serious resource drain. Nutrient deficiency risk: rapid water loss without sufficient mineral uptake may lead to imbalances or deficiency in plant nutrients. So we have studied the advantages and disadvantages of transpiration. These are plant nutrients. Plants require a variety of nutrients for growth, development, and metabolism. These nutrients are classified based on the amount required by the plant. Macronutrient, they are nutrients required in large quantities, more than 1 milligram per gram of dry matter. And micronutrients, these are the nutrients required in small quantities, that is less than 1 milligram per gram of dry matter, but they are still essential. Here the essential elements are carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, iron, manganese, zinc, copper, boron, molybdenum, chlorine, and nickel. Here the functions are already mentioned here. Nutrient deficiencies symptoms in plant, that is occurs due to nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and iron. Here the deficiency syndromes and the deficiency symptoms are also there. Thank you, my dear students.